All right. Okay. Well, welcome everyone. I'm glad to be here virtually and to kick off this meeting. So yes, I'd like to speak today about recent work that we've done. I mean, it's actually work we've done for quite some time, but we're only getting around to publishing it about now. So I'll talk about adaptive absolute binding free energy calculation and how we can apply this methodology to look at fairly complicated binding process. So of course, I suppose everyone in the audience is well aware that alchemical free energy methods are now widely adopted in drug design, and in particular, relative binding free energy methods, using this so-called thermodynamic cycle approach to obtain relative free energies of binding for analogs in cogeneric series have proven quite useful for certain problems in structure-based drug design, such as hit-to-lead optimization. And it's interesting to reflect on the timeline that it took, to go from really the conceptual idea on why this could even work in the framework of thermodynamics to developing the numerical methodologies that actually enable us to do any kind of meaningful calculation on such complex systems. And really decades of academic research that, I would say only in the last 5 years or so have started to lead to more widespread adoption of this technology in an industrial setting. So of course, it's good to be aware that relative binding free energy calculation methods still have somewhat restricted applicability to drug design problem. So you're typically limited to data set of structurally similar molecules with most implementation that are commonly used. So this sort of problem, for instance, you've got this benzotriazine, and you're asking yourself, could I replace this nitrogen here and add this nitrile group? So that's like a good use of relative binding free energy method. But if the question you ask is, I've got this molecule, would this one be a bit better or worse? It's slightly more difficult to tackle that with relative binding free energy methodology, and it's got to do with things like being able to map molecules that are very different topologies. So in a way, it would be much simpler if we could just directly ask for any kind of molecules a question, what is the free energy of binding of this or that, and then use that to rank molecules. So we could imagine that would open up use case for free energies that we don't quite see yet, such as shortlisted compounds on virtual screen, as a filter for de novo design, prioritizing hits you may have from a fragments campaign, for instance, thinking about scaffold hopping, or even binding selectivity studies, and we'll talk a bit about that today. So, of course, many of you know that there is an alternative that could tackle in principle this use case, and that's to use absolute binding free energy methods. And essentially what we do here, if you're not too familiar with the detail, is we still use a concept of thermodynamic cycle, where we've got a molecule X of interest, but then instead of changing it into an other real Y molecule, we make Y an ideal or non-interacting variant of X. So why this is not the thing that everybody uses already? Well, it has a number of limitations. It's quite time consuming to get meaningful reproducible number from this calculation. And it's to do with the scope of the perturbation that needs to be done. And also because of that, you're a bit more sensitive to the quality of your force field. You've got more interaction that are involved in the free energy process, and you can end up with potentially more systematic error. And so because of that, this approach hasn't been benchmarked as extensively as relative binding free energy methods. And then if you look at the literature, there are certainly examples out there, which I would say tend probably to favor slightly lower molecular weight compound than the lead-like space. And we are still trying to work out when these don't work. So we probably see more success on fairly rigid binding site, but there's a lot of open question about when can you robustly apply these methods or not. So we have been exploring this question in the lab for a number of years, and we really wanted to push the envelope of absolute binding free energy And look at a system that we're familiar with, which is the protein MDM2, where not only we're dealing with pretty complex molecules, but also we're dealing with substantial conformational change that are coupled to the binding process. So the MDM2 protein here, the N-terminal domain, has two parts really for the purpose of this talk. A somewhat structured core region and a very flexible, intrinsically disordered lead region here that in apostate can fluctuate between open and closed states. Now, what we find interesting in this system is that if you carry out biophysical measurements to determine dissociation constant for this compound, we can see that, for instance, for this scaffold, natlin 3A, if you truncate the lead somewhere here and make a short variant of this, it doesn't really affect the binding affinity of this compound. If you look at the ratio of dissociation constant between a construct with a truncated lead and a full lead, it's about almost 1. Now, if you take this scaffold here, piperidinone scaffold, we see a different picture. There's a 25-fold preference for having the full intrinsically disordered region present in. And so that's a very interesting question for us. Why does PIP2 bind much better the MDM2 lead construct? So it is something we looked at in previous year using simulation method, and then we came up with some qualitative explanations. So if we carry out enhanced sampling simulation, we get a picture where we see that, for instance, when natlin binds to MDM2, we see that the lead region here in green prefers to stay close and wrap above the ligand and a long helix here. And that's broadly similar to what we see in the so-called closed state in APO simulation of MDM2. For PIP2, we see a different picture. We see in our simulation here, we see that the lead adopts ordered helix term extended motif that becomes a bit more flexible as you go along the extended strand, which is broadly consistent with crystallographic data that can be obtained for this compound. And so that does show that there's quite a different response of this intrinsically disordered region to the presence of these two compounds. So really what we asked here, can we go a little bit further and provide more quantitative insight in the energetics of this process using free energy methods? Now, when you deal with systems of that complexity, you start to hit a little bit the limit of absolute binding free energy methods. And the way we typically do this, normally when we do free energy calculation, we often use so-called static workflows where we plan a calculation, we select a lambda schedule, and then we tend to allocate a same amount of sampling to every window along the schedule, carry out the simulation, collect the data, and do the analysis. Now, I will argue that for absolute binding free energy calculation, this sort of schedule may not be very efficient. And you can see one example here where if you push this sort of protocol, and we're taking PIP2 here, calculating in absolute binding free energy to a short construct of MDM2, you can see the typical profile here between different replicates, independent replicates of the calculation. So you see plots here, the dashed lines are 5 different calculations done. And you can see that if you keep running the calculation for quite a while, we're talking up to about 50 nanoseconds per window. There's about 70 windows in the schedule here. We still have quite some scatter, up to 2 kcal per mole in the absolute binding free energy between replicates. Now, the mean is a little bit more stable, the solid blue line. And so it does show running multiple replicates help a little bit. But you're still looking at, to get somewhere here, to get about half a kcal per mole of uncertainty. In your estimate, you need almost 20 microseconds of cumulative sampling time. So that does limit throughput. Now, if you analyze this a bit more closely, what you find out, though, is that the computational burden is very uneven in this calculation. And so here you can see all the windows in the lambda schedule, and this is an estimate of the uncertainty, the standard deviation across 5 replicates for each window. And there's quite a range. You see there's only a few windows here that contribute most of the overall uncertainty to the free energy change. Now, what that suggests is that using an adaptive approach to carry out the calculation can significantly optimize the efficiency of the calculation. So what we've developed here is a different approach where we carry out simulation for a short amount of time across the replicate, carry out a preliminary analysis, identify which windows are noisy, which one are well behaved, and then allocate additional compute to the windows that do need more sampling. And so you end up with an overall profile sketched here where you allocate more computational effort only to the part of the calculation that requires it. So if you compare now a static protocol versus an adaptive protocol, we can see that now we carry out an initial round of calculation, and that's quite expensive because all the windows need to be processed. But once we got this information, we optimize resource allocation for the next epoch in our adaptive protocol. And then we can iterate that process. And we find that we end up to virtually the same answer than the static protocol, but using about 6-fold less compute time than the static route. Now you have to, there's some additional protocol to use the adaptive sampling protocol here. Essentially, you need to decide on the threshold that you have to set to decide at every round whether you need to extend the sampling or not. And that needs a little bit of tuning. You can see if you set the threshold at a very low value, then you don't gain much because you carry out most windows from one run to the other. So you still end up with actually the same answer, but you haven't saved as much compute time as versus a brute force approach. If you push the threshold too high, what happens is your initial run does most of the cost, and afterwards you only carry out a very small number of windows, and then you end up terminating the calculation too early with a bias in your result here. So you save a lot of compute time, but your estimator is biased. So we find here on the system we investigated that the sweet spot is about 1.1 kcal per mode. So I have no guarantee this would generalize yet. So once we felt we had a way of working out binding affinities for MDM2 ligands at a reasonable cost, we set out to carry out affinity measurements to get very precise measurements of binding selectivity across different constructs. And we felt that was important for this binding selectivity study to really understand the behavior of the lead, because if you just take results from the literature, there can be a bit of variation in the dissociation constant just due to differences in the way the constructs have been done or the way the measurements was carried out. So we used ITC here. So we use Nutlin 3A. We couldn't use PEEP2 because it's not really available, this molecule, anywhere unless you synthesize it from scratch. And Amgen kindly donated to us a related molecule, which is basically a clinical candidate that came out of this piperidinone program. And that's what we used for our selectivity study. So onto our measurements. So first, we verified that for Nutlin 3A, which is a well-known MDM2 inhibitor, that we could reproduce literature trend. And if we carry out our ITC experiment, we see roughly similar behavior in terms of binding to lead or lead short. For M7209, we see extreme potency against the full lead. So in fact, we need to use a competitive titration protocol to be able to work out its affinity. But to lead short, there's a substantial effect, and we can use a direct titration approach. So if you analyze this data and you look at the thermodynamic signature that we observe, we see some interesting trend. If you look at the difference in enthalpy of binding of Nutlin 3A to lead and lead short, there's basically no difference virtually, and same thing for the entropy of binding. We see overall about 1.5-fold preference for having the full lead, which is quite consistent with the literature. In the case of M7209, we see something quite interesting. There's a significant shift in enthalpy and entropy of binding. So the enthalpy of binding becomes less negative when the lead is truncated, and the entropy of binding becomes more negative. And overall, in terms of energetic balance, we see a 250-fold preference for the full lead for M7209. So we ask ourselves, what could be the reason for this behavior? And we carry out simulations to work out the free energy surface of the MDM2 protein with the lead. And we ended up with this pattern here, where we find there's a dominant closed state and different ordered state here, which correspond to what we've seen in crystallographic studies for PIP2. If you integrate this free energy surface, you can roughly work out that the closed state is about 70–80% of the ensemble, whereas the ordered state is about 5%. So the ordered state can form in ApoMDM2, but is intrinsically less stable. And so with that, we can now carry out our adaptive ABFE study on different states of the lead, either the truncated construct, the closed or the ordered lead, and work out standard free energies of binding for different compounds. What we find is that for the case of nuclein 3A, we see preferential binding to the ordered state by only about half a kcal per mole, whereas in the case of AM7209, there's a big gap of about 3 kcal per mole that favors the ordered state versus the closed state. And so if we put everything together, we can propose this picture where the ordered state in MDM2 exists in ApoMDM2, but it's intrinsically unstable. In the case of nuclein 3A, we see stabilization of both states. Ordered state is a little bit more stabilized, but that's not enough to compensate its intrinsic instability. Whereas for AM7209, there's a strong preference for the ordered state, can shift the balance, and the ordered states become more populated. And we can see that the ABFE method gives us a way to tackle this complex problem. It gives us a design strategy. We can aim, for instance, to maximize the gap between the calculated free energy of binding between different lead states if the goal is to find compounds that can order the MDM2 lead. Okay, so if I conclude then, I've talked a little bit about adaptive sampling, how I think it has potential to really improve and decrease the cost of absolute binding free energy method. There's certainly a scope for a lot more optimization, and that's something we're working on at the moment. I think it should allow us to start to be creative and think about pretty complex binding process, and maybe start to think more about how we can use protein disorder to our advantage when we design compound. And I'd like to point out there are methodological effort needed for ABFE methods, and I'm going to point out one glaring discrepancy between our calculation and experimental data. We find that we systematically overestimate absolute binding free energies. So that needs investigation. And I'm happy to stop here and answer any question, depending on what the host tells me.
Yeah, great. Thank you so much for a very interesting talk. We have a first question. I will ask it, and I will also copy it into the chat, so it might be easier for you to read. So the question is, how do you separate entalpic from entropic contributions to the free energy in the context of absolute or relative binding free energies?
Okay, so short answer, we don't do that in the calculation. So in the ITC experiment, that's something you get from the measurement. It would be nice to do that from in the simulation, but it's not straightforward. Primarily because the free energy of binding converge more faster than the enthalpy or the entropy of binding. And I'm sure Mike Gilson would have a lot to say about that.
We have a second question from Lina. Hi, Julien, nice talk. You didn't mention the charge state of your PIP2 ligand. Did you use the anionic form for the absolute binding free energy calculation?
Okay, yeah, good point. So the piperidinone are charged in our calculation, which is different from nuclein. Which are neutral in our calculation. No, we don't correct for that. We thought, could that be a reason why we have this discrepancy with experimental data? We don't actually think so, because when we look at, we can break down this free energy of binding into a contribution from the charge and the Lennard-Jones term, and we see that the overestimation seems to come from the Lennard-Jones term. So short answer, we don't correct for net charge effect in this particular study, but I don't think that explains the discrepancy with the experimental data.
Great, thank you. So I have a question myself, actually, which is about, you mentioned that if you're trying to tune your selection criteria for your adaptive sampling, you end up having some biases with your estimator. I didn't quite understand where that comes from.
Yes, so I think one way to understand that is if you look at this profile where you see the scatter in uncertainty for each window in the lambda schedule. If you set the bar very high, say 0.2 for the sake of the argument, then essentially very early on after your first epoch, you're going to discard all these windows. You're not going to do more sampling. And of course, the results do improve over time for all of this. So you end up neglecting too many windows. So you get all these windows that contributes a little bit of uncertainty, but if you neglect too many of them collectively, that will create a drift from the ground truth.
I see. I think we have time for one more question. How about this one? Hi, Julien. The talk was interesting. Can adaptive absolute binding free energies calculations be used to understand conformational changes in an enzyme? If yes, how?
Yes, I would imagine. I mean, in general, of course, I mean, conformational change in general, I guess in the example we discussed, the conformational change involved a disordered region. I don't see why not. That could not be the case.
Great, thank you. So, Julien, there's a couple more questions left in the QA, so maybe you can type answers to them while we move on to the next speaker. I guess thanks again for a great talk, and I'll do a weird clap because we can't actually thank our speakers properly.
Thank you, Tony.
And I would like to ask Hannah Baumann to bring up her slides next. She's going to talk about binding free energy calculations, sampling challenges, non-equilibrium switching, and alternate approaches. The floor is yours,